Thank you, William. I would like to thank the organizer for inviting me to give a talk here. Today, I will talk about the novel non-coding RNA and big data. So our lab is an RNA bioinformatics lab. So I will talk about the non-coding RNAs here. I would like to start my talks from a small story. So a couple of days ago, I was having a dinner with some speakers outside the Tsinghua. Some of them are from a well-known hospital, also in Beijing. It's called Tiantan Hospital. It's about an hour away from Tsinghua. It claims to have the largest amount of the neuron diseases. They have lots of CT and MRI images for brain tumor. Okay, they told us they have developed a very good AI system, and they are going to release this system in a few years and replace the human doctor in Tiantan Hospital in a very few years. So we are asking, what is this system? Actually, they have already advertised this system. They hosted a competition called Chain Competition in the summer of this year. It's AI vs doctors, actually 10 human doctors. The result is very striking and also expected. The AI is 21% higher than the human doctors in terms of accuracy. And also the costing time is half of the human doctors. So we are asking, how can a hospital develop an AI system which is so good? And they told us, actually, the AI system in the hospital is not smart at all. Actually, it's very stupid in terms of algorithm. The reason this AI system is so good is because of the training the AI system with the largest database of this image data. They are having the largest population and samples in the brain tumor, particularly in China. So this brings out my question about here. So what AI mainly relies on? So many people, including me, believe that it relies on, and simply relies on, big data. Okay. So the big data related to cancer include two major types. One is image data, like here. The other one is genomics data. Genomics data, the processing and analyzing of genomics data is much harder than image data, because for image data, you can easily transfer the algorithm from face recognition, which is well developed in the new computer science. But genomics data is much complicated. So today I will talk about the RNA sequencing data. This is one type of the genomics data which my lab specialized in. So I will talk about application of novel non-coding RNA data on liquid biopsy of cancer. I will introduce the background first. First, I will talk about what is non-coding RNA. So in the central dogma, the DNA will be transcribed into RNA, and RNA will be translated into protein, right? And the coding RNA is messenger RNA. It's coding for protein. The non-coding RNA has only two types, ribosome RNA and tRNA. This is what we have learned in high school. But in the last 15 years, we know that the RNA world has been expanded. Including ribosome RNA and tRNA, we have RNA-SP, ribozyme, riboswitch, extra-cellular RNA, and many other types of non-coding RNA, especially some long ones like hot air and MLAT1. They are highly expressed in cancer. So we know that the messenger RNA is only one branch of the RNA world. Many non-coding RNA exist. For the novel non-coding RNA, you can see the amount of them are very large. We know that 75% of the human genome has been transcribed, and only 2 to 3% are for protein coding. The other 0.01% are microRNA sequences. What else? So most of them are longer ones. When we talk about the function and disease-associated regions, we say it's GWAS SNPs. 12% of the GWAS SNP is in coding region. However, about 90% are in non-coding regions. Many of them are non-coding RNA and also enhancers. Actually, non-coding RNA has been used before as biomarker a while ago. For example, HULC for liver cancer, PCS3 for prostate cancer a while ago. We also have know the function and mechanism of some of the oncogenic and tumor suppressor non-coding RNA, like this table. For example, molecular 1, this is called ncRNA P21. There are also non-long coding RNAs related to cancer. The mechanism is well studied. However, too many non-coding RNAs are not known. In the database of ENCODE and TCGA, they included about 15 and 58,000 of non-coding RNAs. Only about 200 of them have been well studied. We know the function and the mechanism of 200. The other thousands of them we are not known. So we hope to characterize the non-coding RNA, these novel ones. We can simply characterize the non-coding RNA by genomic position. For example, someone we call it intergenic, someone we call it antisense. This is a simple way we can characterize non-long coding RNA, but this is not related to function at all. So we are thinking about the other ways. I will talk about 4 things to illustrating the structure of non-coding RNA, so we can know the function of this structured RNA better. So this is a better way than genomic location to characterize non-coding RNA. I will talk about 4 types. The first type is canonical structured non-coding RNA, including ribosome RNA, ribozyme, tRNA, riboswitch. All of them have very stable tertiary structure. You can see here. The second type is microRNA. Although the final product of microRNA is very small and single-stranded, the precursor has a very strong helping second structure. So this is the second type. MicroRNA also has structure. The third type is non-long coding RNA. There is no single structure of non-long coding RNA for nowadays, but we believe that it must have some structured domains. This is from a Nature review. So by the nature of RNA, it is a very ideal material for sequence-specific recognition. We call it a secondary structure. You can best pair it. You can form secondary structure. So this is a structure we can imagine for long coding RNA. They have domains. The fourth type, which is always missed by many people, but this is in textbook, microRNA introns. The microRNA introns is called lyrate. When we call it lyrate, we are thinking about it is a line, right? That's why we call it lyrate. But it is not a line. It is structured as this. This is a secondary structure. This is a tertiary structure. This is published in Nature in 2014. We know that even the intron of messenger RNA, it is structured. So I'm using these 4 types of non-coding RNA to illustrating structure information, and structure data also need to be included in our analysis. So this is one way I'm explaining why the genomics data is so complicated. So we can say non-coding RNA domains are likely structured. We can use it to separate different types of non-coding RNA, like this. Okay, I will talk about the second part. Because the structure of non-coding RNA, we are expecting some part of this non-coding RNA could be stably exist in blood because it's protected by protein or by its own structure. So we are going forward to look at this RNA. Actually, in RNA, in liquid, in blood, there is an organism called, there is an organelle called exosome. The exosome is a membrane-like circle. It protected some proteins and also RNAs. It includes microRNA and also many other long RNAs. The type of this exRNA sequencing could be separated into cell-free, exosome, microvehicles, and RNP. So we can have 4 types of RNA sequencing data for the blood RNA. And it also, we can sequencing the small ones and the long ones. This is a very advanced technology nowadays. So the biomarkers for liquid biopsy, usually we talk about ctDNA and exRNA. Usually exRNA are missed because it's very difficult to study it. There are some negative view and positive view about this liquid biomarker. For example, the negative view. In a very famous review this year in early 2018, it's called circulating tumor DNA analysis. So lots of experts analyzed the recent papers, the past papers of all these years, studying about ctDNA. The conclusion is very negative. You can see they claims there is no evidence, little evidence for the ctDNA. Usually, there's no evidence of ctRNA in this paper, right? This is a negative view. But also, we have some positive view and new promises. In the, I think it's February, and also the, I think it's August of this year, published 2 papers. The first paper is about cancer, ctDNA. They have developed a technology called CancerSeq. The other one is about cfRNA and exRNA. It's about RNA. So the first one is about DNA, the second one is about RNA. The reason the first one has been successful on DNA is because of 2 things. One is deep sequencing. They deep sequenced all the ctDNAs, and they also included the proteins, add proteins. The other is machine learning. They combined proteins, DNAs, and machine learning, and deep sequencing technology. All these things together, they can achieve very good performance. This is the first thing. The second thing is about these RNA things. The RNA sequencing in blood is not very successful in the last few years. But this is the first successful paper I have ever seen in this year, published in Science. It's not in cancer, it's in pregnant people, in women. So using RNA sequencing data, they can predict the pregnant date and also the measurements to predict the risk of spontaneous preterm delivery. Okay, so you can see from the blood, you can also measure the RNAs. However, in the study of exRNA, most of the study only focused on microRNA, and some of them focused on messenger RNA. We missed the other part, which is the novel non-coding RNA, which is lots of them. So we are developing methods and results. I don't have enough time. I will talk it very fast to introduce our methods. So my lab is utilizing 2 power, sequencing power and computing power. Okay? So in the last 10 years, we have developed many softwares. You can access it from this page called software.ncRNAlab.org. Okay? We have lots of software, and this is a published paper I have been listing here. I only talk about 2 examples. The first one is called RNAfinder. RNAfinder has multiple input, like sequencing data and structure. It uses a supervised model, like many machine learning models, so we can predict the non-coding RNA. And I will skip this ones. The performance is very good. This is before the machine learning. This is after the machine learning. You can see the CDS coding sequence are separated from non-coding RNA very well after the training, after the machine learning. This is the performance of RNAfinder. The application of RNAfinder is in HCC. We published recently. We get some examples, and the performance is being validated by wet experiments and also by TCGA data. Okay, so this is the first example of RNAfinder. The second software I'm introducing is not published yet. It's called exSeq. We are still writing the paper. exSeq is a software to find novel exRNA biomarkers for cancer. So also, we input multiple data like RNAs, sequencing data from blood. And we are using supervised model to predict the cancer, for example, CRC cancer, from the control. Okay, so I will skip these details. The performance is very good. You can see the accuracy is better than a previous payload. It's close to the 19%. This is the AUC, it's about 92%. So after the sequencing, after the machine learning, we can achieve a very good performance of exSeq to predict cancer from control. The future plan. So in the future, first of all, I'm still going to develop bioinformatics software and pipeline for exRNA. So you can see there are many details in this software. The second one, we are going to develop a standardized sequencing protocol for exRNA, also including many library and RNA sequencing. So we would like to set up a database called Dynamic Biomarker Atlas. We need a lot of collaboration, especially many collaborations from hospitals. So I'm posting web page here. You can scan it. If you scan it, you will see our website. So we are looking for collaborators, especially which has samples from hospitals, because we are sequencing lots of samples and build the machine learning model. We need lots of big data. For the last, I would like to thank the listeners again and my collaborators and these hospitals. Thank you for listening.